My name is Gigi Stortz, and it is my privilege to introduce Dr. Feng Zhang on behalf of the RNA and the Lambda Lunch interest groups here at NIH. So Dr. Zhang, as many of you here probably already know, has revolutionized science, not just in one, but in two major ways, by contributing to the development of optogenetics, the technique of using light to control specific neurons, and the development of CRISPR-based tools for genome editing. And he's still amazingly young. So who knows how many more important tools he's going to develop. So just a brief biography. Dr. Zhang was an undergraduate at Harvard, where he worked with Zhang. For his PhD, Dr. Zhang went to Stanford, where he worked with Carl Deiseroff and Ed Boyden on developing optogenetics. He then moved back to Harvard, where he was a Harvard Junior Fellow and worked with George Church as well. Back at Harvard, he first worked on TAL, or transcription activator-like proteins, as tools for genome and epigenome engineering, but then quickly realized the potential of the CRISPR systems found in bacteria and archaea as tools for the editing of mammalian genomes. Dr. Zhang currently has appointments in various institutes and departments at MIT, I won't name them all, as well as at the Broad Institute of MIT and Harvard. As might be expected, Dr. Zhang has been awarded too many honors to enumerate here. I will just mention the NIH Director's Pioneer Award, the Alan T. Waterman Award of the National Science Foundation, and the Gardner Foundation and Tang Prizes, the last two which he shared with Jennifer Doudna and Emmanuel Charpentier regarding the development of the CRISPR-Cas systems for genome engineering. So in the past several years, the Zhang group has contributed much to optimizing CRISPR-based genome editing, and as well as a deeper understanding of the different CRISPR systems in different organisms, and as well as phage defenses against them, and sort of learning how to leverage and optimize all of these tools, which I think will be the topics of today's lecture. So without further ado, please join me in welcoming Dr. Feng Zhang to NIH.
Well, thank you so much for the kind introduction. It's truly a great pleasure to be here. And I've spent the morning here interacting with a number of close collaborators and colleagues and friends, and it's been truly a really enjoyable day here. But it's also really a tremendous honor to be here at NIH and to share with you some of our work. NIH has been the key supporter for all of the work that we have been doing in our lab. And so it's really a tremendous opportunity for us for me to be able to tell you about some of the things that have been done under your auspicious support. So by way of getting started, we have been working on developing molecular technologies for studying biology. And indeed, the study of biological systems and also to address important problems in the world, some of the solutions come from biology. If we just look at the need for improved biological tools, all the way to developing molecular therapeutics and to developing visualization and neuroscience technologies, or to develop sustainable, ecologically friendly and sustainable resources, many of these things are major problems facing us, and the solution to some of them lie in biology. So where does it come from? Some of the solutions come from the enormous biological diversity that surrounds us. If you look at this tree of life, you can see this is where the eukaryotes are, but really if you look at the bacterial cells, they really represent some of the most amazing and most enormous diversity out there. And so needless to say, many of the organisms that live over here have over billions of years evolved very novel and very powerful molecular mechanisms to allow them to survive in different ecological environments. Most of those mechanisms we don't possess, and by understanding and harnessing some of them, we can potentially develop very transformative biotechnologies. So just to give you a few examples that have come out of microbial diversity, the restriction enzymes that fuel the development of molecular biology came from microbial antiviral defense systems. Viral vectors that are sort of paving the way for gene therapy also came from natural sources, all the way to fluorescent proteins, optogenetic proteins, as well as when you look into your everyday household detergent, there are lipases, cellulases, and all sorts of enzymes that are also harnessed from the natural sources. And so inspired by all this, we thought to continue to see, can we explore the natural diversity more and solve important challenges that face our lives and face human health. So one of the problems that we focus on is trying to see, can we develop new solutions to be able to address the ever-expanding list of genetic diseases. Since the completion of the human genome, we now know over 6,000 different genetic diseases that are caused by genetic mutations. And if we can go in and try to reverse those mutations, we can potentially develop treatments for these diseases. Many of them we don't have any treatment for. So the solution to this lies in an area of technology called genome editing. And this is really an area that's been paved by Jim Haber and also Maria Jason. More than a decade, almost 2 decades ago, they reported the observation that if you can induce DNA double-strand breaks in specific sites in the genome, these breaks are reacted by the cell as DNA damage, and they will recruit repair machineries belonging to one of 2 different repair pathways called non-homologous enjoining and also homology-directed repair, and they can allow us to very efficiently make precise alterations to the DNA in our genome. So then, based on this initial observation, many groups have attempted to develop DNA endonucleases that can be reprogrammed to introduce double-strand breaks in specific sites in the genome. So this schematic shows the basic idea. If we can somehow tailor the recognition domain for a specific DNA sequence so that it can recognize any sequence we want in the genome, then this DNA binding domain can be used to bring a nuclease activity to a specific site and make a double-strand break. So you can think of this double-strand break as a cursor in Microsoft Word, except we're now looking at the word processor for letters in the genome. Wherever you can place it, that's wherever you can make the edit. And so several technologies have been developed to make it possible to customize the binding and recruitment of a nuclease. Some of the earlier technologies include zinc finger proteins and also tail proteins. Both of these are protein-based DNA binding domains. So that means these proteins use amino acid side chains to be able to recognize specific DNA sequences. The complexity of re-engineering each one of these DNA binding domains can be pretty challenging. Zinc fingers have individual domains that can recognize 3 bases of DNA at a time, but in order to customize the recognition specificity, it can take quite a bit of directed evolution and engineering effort, and even then it's not guaranteed that we would derive a zinc finger protein that has high efficiency and also can bind very precisely. So one step beyond that was a second system that I participated in developing, and this is a system called TAIL. And so TAIL are from plant pathogens, and they have repeat units that are 34 amino acids long. And so those are these individual color modules. They're more or less identical to each other except for 2 amino acids, and that's these 2. So depending on the composition of these 2 amino acids, that domain can recognize a different DNA base. Because there are only 4 DNA bases, that drastically simplifies the system. It means that we only need 4 different modules, and you can link them together in any combination you want to recognize a new DNA sequence of choice. But this system still has its downsides, which is these modules are very, very repetitive, and if you were to use standard PCR-based gene assembly, it's quite cumbersome and challenging to assemble a desired combination of these repeat units. And so we thought maybe there are even easier solutions to this. And one of the solutions lied in a different system that's also involved in bacterial defense. And so this is a system called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat. And this acronym basically describes the nature of the repeat sequences that are the signature motif of a CRISPR system. These CRISPR systems have repeat arrays where these sort of 30 to 36 nucleotide long repetitive elements are interspaced by fixed length of non-repetitive sequences called spacers. And these spacers are nucleotide sequences adapted or acquired from viruses that had invaded that bacterial cell previously. So each time that virus comes in, these previously acquired nucleic acid sequences act as memory to work together with these protein belonging to the CRISPR system to be able to mount a recognition and immune response against the invading virus. And so if a virus injects its own DNA in, this CRISPR array gets expressed as RNA and processed down into individual RNA guides, 1 spacer per guide. And that can get loaded onto the Cas proteins to form a complex that facilitates recognition of the RNA guide with the target viral DNA. And if there's recognition, the enzyme will get activated and will cleave and form a double-strand break in the virus DNA. That double-strand break, in the context of virus infection, inactivates the virus, but we thought maybe we can transport the system into mammalian cells to be able to introduce precise double-strand breaks in specific sites where we want to place this genome editing cursor. So I learned about the CRISPR system when I first read one of the papers published by Sylvain Moynier, who is a Canadian microbiologist. And so back in 2010, he had reported that if there's a specific sequence in the CRISPR array and it matches a sequence on the virus, the phage DNA, then the enzyme Cas9 will facilitate a cleavage event 3 bases away from the 3' end of the recognition site. And so this implies that this is an RNA-guided endonuclease system. And so if we can harness this system and make it work in eukaryotic cells, then we can have a much easier to reprogram system for achieving genome editing. And so working with a number of students, including Le and also Anne in the lab, we developed the system to be able to introduce Cas9 as well as the CRISPR RNA and the tracer RNA into mammalian cells, pre-programming the CRISPR RNA to recognize the specific sequence on the genomic DNA that we want to edit, so the RNA would hybridize with the DNA, and then Cas9 can facilitate a double-strand break and then stimulate the repair machineries in the cell to allow genome editing. And so since then, we've also worked on, a number of other lab members worked on trying to better understand the parameters that govern the specificity and also the efficacy of the system, and then working also to develop the computational algorithms, which is something that Ben Holmes led. And so that really underlied some of our early work on developing the CRISPR-Cas9 system. But just to summarize that, I want to show you a movie of how the system generally is thought to work. So here you have the Cas9 enzyme, which has been preloaded with RNA guide that's been programmed to recognize a specific site in the genome. By introducing this into the human cell nucleus, this enzyme will search along the genomic DNA and then try to see whether or not the RNA guide is able to base pair with the target DNA site. And if the RNA base pairs, then the enzyme will get activated and will make two cuts in the 2 strands of DNA and forming a double-stranded break. Now, two different repair scenarios can happen. Without a template, the two DNA ends will get rejoined through a process called non-homologous injoining. And this is an error-prone process that can introduce a small mutation that can inactivate a gene of interest. So it's good for knocking genes out. Alternatively, we can activate a second repair process called homology-directed repair. And this is done by providing another piece of DNA template that carries homology to the two broken DNA ends, but in between is a new DNA sequence that we want to introduce into the genome. And through homology-directed repair, this piece of DNA can get incorporated into the genome and then allowing us to make a very precise change within a specific site in the DNA. So that's to facilitate genome editing. But in addition to cleaving DNA using these two catalytic domains in the Cas9 protein, we can also inactivate them so that we can turn this into a generic RNA-guided DNA binding domain, and that's what's shown on the bottom here. The DNA binding domain can be used to recruit specific effector modules to different sites in the genome. You can recruit a transcription activation domain to turn the gene on. You can recruit a cytidine deaminase domain to be able to modify a single DNA base to change a cytosine to a uracil. And also you can bring GFP if you want to visualize the conformational dynamics for a specific locus within the genome. So there are many different ways where this Cas9 RNA-guided system can be used to study genome function. So I thought I'll share with you a few vignettes from some of the work we've been doing recently. And so some of the things that we've been doing to improve the Cas9 targeting specificity, and then how we can apply it to achieve high throughput genome scale perturbations to look at gene function, as well as modeling specific gene mutations to understand their contribution in disease. And then finally, some of the work that we have been doing in collaboration with our dear friend Eugene Koonin here to explore CRISPR diversity to be able to further expand the CRISPR genome editing toolbox. So we'll start with the first one. So we know from many studies, both ours as well as many other groups, that Cas9 can sometimes introduce off-target activity. So besides editing the site that you want to modify, it can also introduce other unwanted modifications. So working with Ian and Ling-Yi and Bernd, three members of my lab, we looked at the way that Cas9 binds to DNA and thought maybe there are ways that we can re-engineer one of the features of Cas9 to make it much more specific. This schematic shows the way that Cas9 would bind to DNA. So gray and the purple and blue or green are the Cas9 protein, and the purple and the green signify the two catalytic domains for cleaving DNA. And the bound DNA is shown here in red, and then the green is the RNA guide. So for this complex to achieve recognition and cleavage, the DNA, which is normally in double-stranded form, needs to be unwound so that one of the DNA strands can base pair with the RNA guide. When that happens, the other strand of DNA needs to be stabilized. Otherwise, it will want to rezip and reform a helix and eject the RNA guide and the protein. So in order for cleavage to happen, this binding event needs to be strong enough. So to make it strong enough, this DNA strand, which is negatively charged, needs to be stabilized by some positive charge on the protein. And so we looked at the crystal structure, which allows us to examine the surface charge density. This is the same crystal structure for Cas9. It's colored in the same way. So rather than looking at the domains, we can recolor it to look at the surface charge distribution. And so here is a different way of coloring it. Blue means positive charge, and then red means negative charge. And so you can see this groove here where that displaced DNA strand is supposed to fall is pretty blue. And so what that means is that there are a lot of positively charged amino acid side chains, arginines, lysines. And so we thought maybe there's too much positive charge. So even when the RNA is not perfectly matched with the DNA, this strand is sufficiently stabilized through excess positive charge that there's very little willingness for this DNA to rezip with the other strand and then eject the complex. And that's how you end up with the off-target DNA cleavage event. So we then examined this and identified a large number of arginine and lysine residues and thought maybe if we convert them to alanine, something that's not positively charged, we can weaken this interaction and then make it possible for Cas9 to become more specific. And so we selected a large number of these mutations, and then through combinatorial testing, we found that some mutations were able to make Cas9 significantly more specific. So here, this is the wild-type Cas9, and we selected a guide RNA that targets VEGF-A. And so here, for the gray bar, we can see editing at the on-target VEGF-A site. From our previous work, we knew that this guide has 2 additional off-target sites, and that's OT1 and OT2. So with the wild-type Cas9, both of these off-target sites were significantly modified. You see almost 10% indel mutation, which is very, very high. Now, after screening a large number of mutants, we identified 2 different combinations of mutations. Both combinations carry 3 substitutions that convert lysines and arginines to alanine. And this ESP-Cas9, enhanced SP-Cas9 1.0, preserved the on-target editing and abolished the off-target editing activity. And also, the second combination also preserved the on-target editing and then was able to reduce off-target editing to the same level as background noise level. And so from this, we knew that through this type of rational design process, we can make Cas9 more specific so that as we develop Cas9 into a human therapeutic, we can make the system safer through these kinds of engineering approaches. So let's switch gear a little bit and look at how we may be able to apply this system to study gene function. So one of the nice things about the RNA-guided system is that the RNA guide can be very easily synthesized. In fact, we can print DNA oligos at very large scale for very, very low cost so that we can program Cas9 simultaneously to edit many, many different genes. So using computation, we design RNA guides that can target every single gene in the genome, and we usually design multiple guides so that there's some level of redundancy so that we can account for a potential off-target editing activity. And then using oligoarray synthesis, we can print these RNA guides in DNA oligo form using inkjet printing. And this is similar to what people will do to make a shRNA library to target the whole genome. We can clone these Cas9 guides into vectors and produce viruses that can then be put onto a population cell at a low multiplicity infection. The reason we do low multiplicity is because we want to introduce 1 virus per cell and not have more than that. More than that will make it very difficult to deconvolve to figure out what is the mutation that's doing the interesting perturbation. With this population of cells, we have a broad array of genotypes. Each cell received a single guide, and so over a large population of cells, we now have every single gene perturbed, 1 gene per cell. And so then with this population, you can apply a selection onto the cell. You can look at, you can ask questions like, what are genes that when you knock out will render the cell resistant to a certain virus infection? Or what are loss-of-function mutations that confer metastatic properties for cancer cells, and so forth. And so we can look at the result of this selection by sequencing the guides that are remaining in the surviving cells, and by mapping them against different genes in the genome, we can then identify a list of candidates that participate in a specific role in the biological function. So we show that you can do this for both loss-of-function screens using the Cas9 nuclease system to introduce indels. But you can also perform gain-of-function screens where using Cas9 to recruit a transcription activation domain, you can turn different genes on and ask the inverse question of what are the genes that you need in order to mediate a specific biological process. So this is all really nice for studying protein coding genes. But what about the rest of the genome? After all, the majority of the genomic sequence don't encode proteins. So we asked the question, can we develop this as a way to study the function of non-coding sequences in the genome? And so two of my former lab members, both of them are now independent, developed a way to be able to develop libraries of RNA guides that target promoter or 3' untranscribed regions of the genome so that we can introduce indel mutations in a saturation way so that we can ask what are the non-coding genomic regions that encode important motifs that are important for gene regulation. So the way Neville and Jason did this is they designed guides that basically target every possible target site in the 5' and also 3' region for a gene of interest. And then using the same library generation method, we print the oligos and then clone them into various libraries. And so then you can take this library of cells that have different perturbations along the non-coding region and then apply a selection. And in this case, we looked at what are genomic non-coding elements that perturb the expression of a specific gene and then drive resistance in the presence of a cancer therapeutic. So this is the gene that we targeted. It's called COL3, and it's a gene that we knew from a previous screen. If the gene is inactivated, then the cell will become resistant to vemurafenib selection. And so we designed guides that tile along the entire 5' and 3' region. Each one of the blue bars you see here is a target site in that genomic region. And so by performing the screen, we looked at the surviving cells, and we saw that there are specific guides, about 7.2% of them, that became enriched after selection. And so let's zoom in a little bit more and see where are those 7.2% of guides. So this is the guide frequency plotted along the length of the 5' and also 3' region. So you can see that there are specific areas where concentrations of the guides are observed that are surviving, that are allowing the cell to survive in the presence of the drug selection. So if you zoom into some of these peak regions, you will see that many of these peaks represent target sites that bear motifs to known DNA binding sites. And so, for example, if you look at this one, this also overlaps with the basically binding site for the YY1 transcription factor. And if you look at a specific DNA sequence, you can see that they carry a particular motif. And so if you go through all these other sites, you see that many of them represent known DNA binding motifs, but some of them also point to novel DNA binding sites. And so using this kind of methodology, we can both study protein coding genes as well as start to map with very high resolution sort of regulatory elements in the non-coding genome. So this is to screen. This is when we don't know what is the gene that we want to study, and we want to discover the specific genetic element that drive this type of biological process. But sometimes we already know what a gene is, or we have a list of candidates, and we want to dive in deeper to understand their function. And so to give you a sense of how we might do that, we have been applying this to study autism spectrum disorder. So about 6 years ago, three really important papers were published. They were looking at de novo coding mutations that are found in autism spectrum disorder patients. And so through these three papers, they identified a number of different mutations that are highly correlated with autism spectrum disorder. So one natural question is, what are the causal role of these specific genes? So are they just based on, are they simply correlated, or does it actually mediate a causal relationship with autism-like phenotype? So taking the list of candidate genes, we use genome editing to try to develop models that allow us to study the function of these specific genes in the context of autism. So we conducted two different lines of modeling approaches. The first one is in vitro modeling based on stem cells, where we can begin with a well-characterized healthy stem cell line and then use the genome editing system to put in the mutation that's found in patients. The benefit of this is that we can generate a mutant cell line that is otherwise isogenic to the healthy unmodified stem cell lines, so that we can conclusively say that the phenotype we observe is due to the specific gene that we modified. We can do the same thing in vivo by generating mouse models, where we take single-cell mouse embryos, inject the Cas9 reagent, and then introduce the same mutation that we try to model in human stem cells. So Neville and Cici are two. Neville is independent now, but Cici is still a postdoc in the lab. They introduced the Cas9 and also the guide RNA into stem cells, and then through FACS sorting, they were able to select out specific clones that carry this specific mutation. And so then we can differentiate these embryonic stem cells into neurons and try to see what happens. So these are neurons that were differentiated based on the stem cells. So we performed patch clamp experiments to see does the physiology of these ESL-derived neurons change when the cells carry a mutation in the gene CHD8. So by looking at the firing threshold, we injected a steady current into these cells, and then we measured the action potentials that are evoked by this input of electrical activity. We found that the wild-type unmodified cells, which is the control, has sort of very nice firing of action potential trains. Each one of these spikes is an action potential. But then for neurons derived from the mutant cells, the firing pattern is much more representative of less mature neurons. And so if you look at the summary statistics, you can see that the number of spikes that are fired by these mutant cells are much lower than the number of spikes fired by the wild-type cells. And so this suggests that maybe these cells are potentially less mature. Maybe they're developing or differentiating a little slower than the wild-type cells. And then we did a second measurement, which is measuring synaptic transmission. And so here, this is synaptic transmission in the cultured ESL-derived neurons for control cells, and then this is the knockout cells. So if you compare the knockout cells with the control cells, you also see that both the amplitude, so how big these transmissions are, and also the frequency at which the transmission is transmitted as spontaneous activity in these cultured cells is both lower compared to the control. So these are the summary stats. So you can see both the rate and also the amplitude are also reduced. And that is consistent with the slow differentiation or slow maturation phenotype that we saw with the current injection experiment. And so this provides a way to start to link physiology phenotype with specific genotype that are observed in these human genetic studies. So it's hard to ask, it's hard to examine behavior in specific neurons. And so we also generate mouse models. And so Randy and Ian took mouse embryos and then designed the CRISPR reagent to be able to also modify the CHD8 gene in these mice. And so similar to human patients, we generated heterozygous loss of function CHD8 knockout mice so that we can study what happens on the behavioral level. So there are a number of different hallmarks of autism, sort of behavioral hallmarks of autism. And so we ran the mice through a number of different behavioral tests. The first one is to look at both sociability and also social novelty seeking. The way this works is as follows. So we have a 3-chamber box. There are 2 side chambers and the middle chamber. We put in one of the side chambers a mouse, a container that has a mouse in it, and then the other chamber we put in an empty container. And so we put a subject mouse in the middle chamber, and then we measure the amount of time that this mouse interacts with one or the other jar. And so if the mouse is sociable, one metric used to measure that is to see how much time this mouse interacts with the jar that contains another mouse, and how little time it spends with the empty jar. So this is to measure sociability. The second test is to measure social novelty, where we have one jar with a mouse that has been previously exposed to the test subject. And then in the other chamber, we put a mouse that's novel, so it has never been exposed to the test subject. And then we measure the amount of time that the test mouse spends with the familiar mouse or with the novel mouse. So this is what we saw. So on both of these metrics, sociability and novelty seeking, the CHDA knockout, heterozygous knockout animals had reduced sociability as well as reduced social novelty seeking. So this is just one of the many behavioral hallmarks. So the second one is to look at anxiety. And so here what we saw is that if you put a mouse in an open chamber, so this is just a square box, and then there's a camera on top of it so you can monitor the path that a mouse walks in the box. And here is the plot showing the pattern that the mouse is able to walk around. The wild-type mouse spends time everywhere. It roams around the center and also the surround, basically everywhere in the box. But if you look at the CHD knockout mouse, it only stays in the perimeter of the box, and it really doesn't spend very much time in the center of the box. And so this is a way to measure the level of anxiety that a mouse might be exhibiting. And so if you quantify this, the amount of time spent in the center versus the surround, you can also see that the CHD8 knockout animals spend far less time in the center than the wild-type mouse. The third hallmark that we examine is ability to learn repetitive tasks or motor learning. And so here, this is a behavioral paradigm called rotor-rod test, where there's a rotating rod, and then we put the test subject on the rod, and then we measure how much time it takes before the mouse falls off of this rotating rod. The longer it stays, the better it's able to perform this motor repetitive learning task. And so this is over multiple trials, and you can see for both the wild type and the knockout animal, the performance improves. So it takes longer and longer for the animal to fall off. But the CHD8 knockout animal is able to learn so much faster. And so this is another indication that there is some behavioral difference in these CHD8 knockout animals. So after seeing all these behavioral differences, we wanted to know what's happening in the brain of these animals. And so we dissected out tissue from different regions of the CHD8 knockout animal brain, and we compared it with the wild type. And in one of the brains, called the nucleus accumbens, we saw that the expression of both NR2A and also NR2B, two NMDA receptors, the expression level was elevated compared to the wild type. And so this prompted us to focus in more on the nucleus accumbens to see is CHD8 mediating all these, or CHD8 knockout in the accumbens mediating all these behavioral phenotypes that we saw. So to test that, we turned back to the CRISPR-Cas9 system. So rather than making a global knockout animal, now we build viruses to be able to deliver the Cas9 reagent into a specific region, such as the nucleus accumbens in the mouse brain. We had previously built a transgenic animal that has a Cre-dependent Cas9. So we then designed an AAV virus that carries both the Cre recombinase as well as a guide RNA targeting CHD8. So by injecting this virus into the nucleus accumbens, we're able to selectively transduce the nucleus accumbens cells, express Cre, activate Cas9, and then use this guide to knock out CHDA selectively in the accumbens neurons. So we can do the injection quite precisely. This is injection of GFP, so you can see that there's pretty localized expression in the accumbens, and if you inject in the dorsal striatum, you can get specific expression in the striatum and not in the accumbens. So we did this region-specific injection to see is CHD knockout in the accumbens alone able to mediate similar behavioral changes. So here we found that on the rotor-rod test, knockout of CHDA in the accumbens was able to sort of also increase the learning performance, whereas knockout of CHDA in the dorsal striatum, so a control brain region, or injection of a guide RNA that doesn't target the mouse genome, neither of those improved the learning performance on this behavioral task. But when we tested the accumbens knockout on the social interaction or the anxiety task, we didn't see a significant change in behavior. And that likely suggests that maybe other brain regions are involved. And so CHG8 is playing a role in other brain regions to regulate those other behaviors. So this is something that we're continuing to do to try to better understand these genes and their roles in the autism spectrum disorder. And hopefully by studying several different genes, we can try to identify converging mechanisms, maybe something a little downstream from these different genes that govern these different autism-like behaviors in mice. So finally, just want to share with you some of the work on exploring CRISPR diversity that we're also very excited about. And part of that has to do with the diversity of CRISPR systems. So CRISPR-Cas9 is probably the most famous version of CRISPR systems, but there are actually many other CRISPR systems that exist out there. And so we've been very fortunate to be able to collaborate with Eugene Koonin here at NIH to start to explore this CRISPR diversity. But let me tell you what these different classifications are. Broadly speaking, we can divide the CRISPR system down to two classes. There's class 1, which use multiple proteins together to form a large multi-subunit complex to carry out the RNA-guided recognition and also the downstream defense function. Class 2, which is the class that Cas9 belongs to, is much simpler. It uses a single protein along with the RNA guide to carry out the recognition and also defense function. And so we thought maybe we can explore these single component class 2 systems and see are there other systems out there that are potentially quite powerful. And we can harness those to develop new biotechnologies. And so working with Eugene and his team, including Kara and also Kasia, who's a collaborator at Skotech in Russia, we use computational methods to be able to search through bacterial sequences to see are there other class 2 CRISPR systems. One version of the search works this way. So by taking Cas1, which is one of the most well-conserved Cas proteins, it's involved in acquiring immunity for these CRISPR systems. We can do blasts and identify all of the contigs that carry the Cas1 protein. Because CRISPR systems are encoded in bacterial operons, so there's a strong likelihood that the nearby proteins are also related to CRISPR function, so you can identify all of the open reading frames nearby. And then for each open reading frame, you can try to annotate it as either something that's similar to what's known before, or as a completely unknown sequence. And so naturally, because we're interested in finding unknown genes, focus on the unknown sequences. And then for each unknown protein, then there are several criteria that would be applied to try to see is there a strong likelihood that this candidate represents a novel CRISPR effector protein. The first one is whether or not this unknown protein represents a whole family of proteins. If that's the case, then that really signifies that this is an evolutionary conserved family of proteins. Second, do most members of the family co-locate with a CRISPR array? And if that's the case, then we know A is evolutionary conserved, so it's important, but 2 is also a conserved CRISPR protein because it's often found next to a CRISPR array. And so by applying this type of metric, we're then able to identify a large number of different candidates. And this is probably the most up-to-date census of the CRISPR system. So there are two types of DNA targeting system. There's the type 2, which has Cas9, and then type 5, which has CPF1, or it's been renamed to Cas12, and also all the other related family members. And then there's the type 6, which represent RNA-guided RNA targeting systems. And these are called Cas13. There's Cas13A and also Cas13B, 2 different types of targeting systems. So we've been studying various members of these systems, and I want to tell you about some of the work we've been doing to try to understand what Cas13 might be doing. So 2 of my graduate students, Omar Abuday and also Jonathan Gutenberg, focus on trying to understand what C2C2 might be doing. working with the natural organism is pretty challenging. So working with Leptotrichia shahi is difficult to culture, and also it's difficult to be able to genetically manipulate them. So instead of working with the native organism, we took the C2C2 locus, so all of the CRISPR genes as well as the CRISPR array from Leptotrichia shahi, and put it into E. coli, something that's much easier to manipulate. Then we did RNA sequencing. So by examining the expression of RNA, we saw that a CRISPR array is not only expressed, but it's also processed into the individual mature CRISPR RNA guides. So that's a very good sign that suggests that maybe this is an active CRISPR system. At least the CRISPR array is processed into the mature form. So based on computational prediction, there are HEPN or heparin domains that are found in these C2C2 proteins. And these heparin domains suggest that this might be an RNA targeting enzyme rather than a DNA targeting enzyme. And so to test that hypothesis, we then designed a screen to see whether or not C2C2 is able to mount immune defense against RNA virus infection. So we chose an RNA virus called MS2. This is a small virus, so the genome is only 3,800 bases long. And so we can design RNA guides that tile along the entire length of this virus's genome. And so then we can synthesize it just like the way we build a CRISPR guide RNA library for screening purposes. And then we can clone each one of these guides into a vector that carries the rest of the C2C2 system. So each plasmid carries the C2C2 system as well as the RNA or a C2C2 guide that targets the MS2 virus. So then we can transform this plasmid library into E. coli. And now we have a library of E. coli. Each cell carries a single target sequence against MS2. And then the whole library targets every possible position within the MS2 genome. So if C2C2 is an RNA-guided defense system against RNA virus, then by putting the MS2 phage on this population, some of the cells should survive. And so if we see cells survive, then we can reasonably think that there is RNA-guided RNA interference against this MS2 virus. And then furthermore, if we look at the spacers or the guide sequences in the surviving cells and analyze any sequence motif, then we can use that to understand targeting requirements for this C2C2 based system. So we did this experiment, and fortunately, we saw that some cells did survive. And so that got us excited, and we thought maybe this indeed is an RNA-guided RNA defense system. And so by analyzing all of the guide sequences in the surviving cells, we found that right next to the target site, which is called protospacer in CRISPR jargon, there's a very simple motif, which is a single base. It just has to be either a C or A or a U. And as long as you have that, then the C2C2 system can successfully mediate defense against this MS2 phage. So now we thought to see how well can we use the system to knock down specific RNAs in bacterial cells. We designed a different experiment. Now there are two plasmids. One of them carries the red fluorescent protein, and then the other one carries the C2C2 system and a specific RNA guide designed based on that C, A, or U motif to target the RFP mRNA. And so by transforming E. coli and then measuring the level of red fluorescence, if there is degradation of the RFP mRNA, then by flow cytometry, we should see a decrease in the red fluorescence. And so that's what we saw. And so this is the control, and here is a negative control where we designed a guide that's not flanked by a C or A or a U. So this is flanked by a G. And this one has 100% fluorescence. And then for the three guides that are designed with the right targeting motif, each one of them has significantly reduced fluorescence. So this suggests that we were able to target the RFP mRNA successfully. And just to make sure that we're not targeting the RFP expression plasmid DNA, we then designed 3 guides that target the template strand of the RP gene. So this is targeting the DNA. And here, we were not able to reduce the red fluorescence. And so this shows that it really is an RNA targeting system. So this is very interesting. We now have a system that we can reprogram with a specific RNA to target other RNA species. But there's something more that's interesting about this system. And that is, in addition to targeting the RNA that's bound by the RNA guide and the C2C2 enzyme, upon recognition, the C2C2 enzyme can get switched on and become a non-specific promiscuous RNase in solution. And so we identified this by designing this experiment. So here is the target RNA, but we don't label it. So even if this RNA is degraded, we don't see it when we run a gel. And here's the RNA guide. So in addition to these 2 RNA, we put in a 3rd piece of RNA that is not recognizable by the RNA guide, but we label this RNA. And so if this RNA gets cleaved, then we know that we can visualize it by looking at this guide. And so by running this experiment, we found out that indeed this RNA triggers the cleavage of this non-specific RNA. And so this is the gel that shows that. So here are 2 different target RNAs, so 2 different versions of this RNA. And then whenever this RNA is present and is targeted by the guide RNA, we can see degradation of the RNA. But if you withdraw this target RNA, so if this RNA is not present, so it's only C2C2 and this CRISPR RNA, then there's no degradation of the input RNA. And so what this showed is that C2C2 really has additional promiscuous activity that allows it to cleave RNA in the vicinity beyond what is bound at the target site. So this particular feature has potential biotechnological uses as well. And so using this, we were able to develop a way to detect infectious agents, such as RNA-based viruses like Ebola and Zika, or to detect various bacterial pathogens, even in the DNA form. And the way this works is a process that we called SHERLOCK. It's a 2-step process. The first step of the process is an amplification step that uses isothermal amplification, so it's very easy to perform. And so whatever it is, whether it's input DNA or RNA, we can amplify it, and then use T7 transcription to turn that into RNA. So that's the material that we then apply C2C2 and the detection RNA to try to identify. To read out the activity, what we do is, in addition to incubating this RNA, amplified RNA pool with C2C2 and the guide RNA, we also put in a reporter, which is a commercially available RNase reporter called RNase Alert. And this reporter has both a quencher and a fluorophore attached to the 2 ends of the RNA. And so if C2C2 recognizes the target RNA and cleaves the target RNA, it will also get switched on so that it can cleave any other RNA in the solution. And so that means it can also cleave this RNase alert reporter. When that happens, the fluorophore and the quencher are released from each other, and you get a fluorescent signal. C2C2 in this reaction provides additional amplification because each molecule of the detected RNA will switch C2C2 into this non-specific promiscuous mode, and that C2C2 can cleave many thousands of copies of the RNase reporter, so you get amplification of the signal. And so using this, you can read out the presence of this specific RNA by just looking at the fluorescence emission. And so what we found is that this system can work pretty well. In fact, the SHERLOCK system, which is the C2C2 and the guide RNA, can be freeze-dried onto a sheet of filter paper. And that's really nice because you can store it very easily, and you can also transport very easily without requiring refrigeration. And the whole process works very quickly. So this is about 20 minutes or so to be able to perform the amplification. And then to perform detection, it's another 20 to 30 minutes to be able to read out the signal. So in less than an hour, you can perform this detection assay. And with optimization, you'll be able to get this even shorter period of time. And the detection works very well. So when we designed guides to detect Zika sequences, we can detect it with very high sensitivity down to the sort of 10 nanomolar range. And so this is a level that we need to get in order to detect early stages of this virus infection. And also, with optimization, we also show that you can get it down to even a single-digit nanomolar range, which is about 1 molecule per microliter concentration. So this is for RNA detection. This is RNA sequences with Zika or dengue. You can also use it to detect DNA sequences. So we amplify the DNA and then use T7 transcription to convert the DNA into RNA. And so here, this is with sort of clinical isolates of bacterial culture, and then we run through the same process. And then for E. coli gDNA, we can detect specifically using a E. coli specific RNA guide and not anything else, so the background is very low. And also for Pseudomonas, we simply switch to a Pseudomonas specific RNA guide, and we can detect that. And also the background for other bacterial species is also very low. And so using this kind of system, we can very rapidly and also sensitively detect specific strains. And you can change the test to detect anything you want just by giving it a new CRISPR RNA guide. And so this provides a very efficient and easy diagnostic system. So this really highlights how there could potentially be many, many more powerful biotechnologies that are awaiting discovery in the bacterial diversity. And so if you just look at the amount of bacterial genome data that has been accumulating over the past 10 years or so, you can really see there's an exponential growth in the amount of microbial genomic data. And so as we develop better capabilities to be able to sequence and also to analyze them, there's very likely additional tools to be discovered. And I think in the coming years, there will be many, many new biotechnology that can potentially make our lives and human health much, much better. So that's all I have prepared for today, but I just want to take a moment to acknowledge my team at Broad and MIT, and also all of the collaborators that we've been so fortunate to work with. And most specifically, Eugene, who has been both really just a tremendous knowledge base and just a really genius of a bioinformatician that we've been very lucky to work with him and his team to study the CRISPR diversity system. And also for the CHD8, we were able to work with one of the experts in autism modeling, Guoping Feng at MIT. And then for developing the diagnostics, working with Jim Collins and Deb and Pardis, who are experts in virus and bacterial infection. And so really being able to work with a world-class set of collaborators has made it possible for us to do all this work. And then finally, just want to acknowledge all of the funding agencies, both at NIH as well as some of the private foundations, individuals that made the work possible. Thank you very much for your interest in our work. So, happy to answer any questions.
Yeah, please use a microphone if you have questions.
Well, thank you so much for such a brilliant talk. We really enjoyed that very much. So recently, you must have noticed that there's a new publication in the Nature Method to show that there's the non-specific mutation and that introduced into a coding sequence that is through the CRISPR process. But that paper also that received the criticism that about some error. Could you provide your comment, and also that what's your opinion about that? So how to avoid the off-target effect of the CRISPR?
Sure. Yeah, that's a good question, and it's an important question. So recently there's a paper that reported there may be thousands of single nucleotide variations that are introduced by the Cas9 system. So we and also many of the labs that I have worked with have not observed that effect. And if you look at a paper a little carefully, you may find that the way that they made the comparison may not allow for very conclusive determination of those single nucleotide variations. The experimental mice that they injected Cas9 into is of a different genetic background as the wild-type mouse. And so given that strain to strain can have quite a bit of SNP variation, it's possible that these SNVs that they reported to be caused by Cas9 are actually due to background differences between these different strains. So some more work needs to be done, and I think it's something that highlights how that specificity is really an important issue and something that we need to work on. In terms of actual experiment and avoiding off-target effect, I think there are a number of things that you can do. The first is to computationally look at the sequence you're designing and make sure that it is as different as possible from the rest of the genome so that you minimize off-target activity. Second is to use these enhanced versions of Cas9 where we introduce mutations that allow them to become much more specific. And then finally, the third thing is to limit the exposure and the concentration of the enzyme that you're introducing into cells. So rather than putting a plasmid that can express thousands, if not tens of thousands of copies of the enzyme in a cell for a long period of time, you can introduce the protein RNA complex, the RNP, into those cells, which will be the dose is much easier to control, and also it will not last for as long. And so if you do all those three things, I think you can get the specificity to be quite high.
Yeah, and same question very quickly. And so is it possible to develop the CRISPR system don't require PAM sequence?
That's a really good question. So the PAM sequence is a small motif that's right next to the target site. And it's been found through a lot of experimentation that the PAM is what Cas9 uses to initially latch onto DNA and to initiate unwinding. And so it's possible that maybe you can design a version of Cas9 that has sufficient affinity for generic DNA sequence, not for a specific base sequence, that can still allow the unwinding. But even if that's not easy to do or hard to get at, it's possible to design Cas9 that requires shorter PAM sequences, maybe just a single nucleotide. And then you can have 4 different versions of Cas9 so you can cover the whole space.
Thank you.
I have sort of a more general question. So you focused most of your talk on genome editing, right? And the ability of Cas9 to cut DNA, possibly to introduce or to correct mutations. Mm-hmm. But it seems to me that the promise of the system is much broader than that, right? Mm-hmm. I mean, you can basically bring to a specific region of the genome an entire regulatory machinery, right? So if you think in terms of impact on biomedical research or new therapy, maybe the broader impact would be on regulating, for example, transcription in complex diseases. So I was wondering, what were your thoughts about the use of those systems beyond simply correcting mutations?
I think you make a very good point. Certainly, being able to cut DNA is only one of many ways in which Cas9 can be engineered into a useful tool. And so there are folks who are using Cas9 as a nuclease to develop human therapeutics. But then there are many new systems that are being reported too. For example, you can use Cas9 to recruit a transcription activator to turn genes on. You can use it to recruit crab domains to be able to repress epigenetically a specific locus. You can also recruit cytidine deaminase to be able to change a specific cytosine to a uracil to make a C2T mutation. You can also use it to recruit methyltransferases, so you can cause methylation, also epigenetic modification. So all of these different things, I think, are very promising. And I think there are many groups that are working actively to get each one of these modalities to a high enough level of efficiency so that we can use it in these kinds of application contexts, including for human therapeutics. For long-term expression applications, like to turn a gene on or to turn a gene off, chronic expression of a bacterial protein in human cells may pose challenges. So these things may get processed, these Cas9 proteins may get processed into small peptides and present on cell surfaces and cause inflammation. But those are things that are also being actively pursued and studied by several groups. So I think it's very bright at the end of the tunnel. There are many different applications, and I think each one of them will certainly have huge impacts.
Hi, for your CHD8 mice, have you done histology to look at neuronal morphology or changes in neuronal populations within the brain that could explain the changes in behavior that you see in your behavioral studies?
Yeah, that's a really interesting question. So we have done some work to look at that. So for example, we have found that the nuclei of these CHD knockout cells have a different size compared to the wild type. And also anatomically, there may be differences which we still need to analyze. So certainly there are changes to the cells. Yeah.
Thank you.
Thank you. Okay, thank you.
In the interest of time. So please join me in thanking Dr. Feng Zhang one more time for a wonderful seminar. I want to remind people who are interested, there's a brief reception next door in the NIH library. And it is my understanding that this is the last WALS lecture for the 2016-17 season, so look forward to more great talks next fall.